Assalamu alaikum. Assalamu alaikum. Now we are going to work on the discriminant analysis. Uh, we will be carrying out this discriminant analysis for the identification of the species of gastropods. Basically, this is a test through which we can confirm, if you're using it for the sake of taxonomy, we can confirm that whether the discrimination which has been carried out among different classes has been carried out correctly or incorrectly. So here's the data which we have pasted from our Excel sheet onto a Minitab page. And the data shows that there are sampling units, and then there are certain characters, and then there are species which have been indicated in the last uh, column. So the point is that we have generally when we carry out sampling, we have different sampling stations, and then we do have different sampling samples, and then we do have different sampling units. So basically, three terms are very important when we are carrying out the sampling. Number one is sampling station, then the sample, and then the sampling unit. Sampling station, for example, if we have carried out um, our sampling from University of Agriculture of Islamabad or from um, Forest Research Institute, then it means that that particular area would be our sampling station. And then we will divide that sampling station into different samples. For example, we may have certain botanical gardens or fish farms or agricultural lands in those particular areas. So those areas would be our samples. And then these samples will be further subdivided into sampling units. And these sampling units will, in our case, these are basically our microhabitats, because our species is not so mobile. Especially the gastropods are very uh, they don't move around very much. So we generally consider microhabitats, um, for example, just under the log or under a plant, or certain areas where they have a micro niche. So on the basis of that, we make our um, data. We take the data of the different specimens from those different sampling units, and then we calculate, or we do the morphometry of those particular characters using our tools. So these are the different characters of the gastropods, that is shell height, shell width, aperture width, body whorl length, spire length, number of whorls, shell coiling, peristome, umbilicus, shape, color. Then we have a very important ratio which is generally considered for the identification sake, that is shell length over shell width and aperture length over shell width, on the basis of which we have identified our species, that is Macroclamus indica. And then we, from different areas, we found three different species. That was, one was Macroclamus indica, the other was Xeropicta candaharica, and the third one was Zooticus insularis. We can see all these three species in this data. And then we have Zooticus insularis. So there are multiple specimens from various um, some areas with different sampling units. So now we want to identify, want to understand that whether we have done the correction or identification on the basis of correct parameters, or the parameters which we have used have been have been utilized very correctly. For this sake, we will be going to the tab Stat, and then Multivariate, and then discriminant analysis. What are the groups? We have species will be considered as groups. And what are the predictors? All the predictors are generally all those which are which have a quantitative number. You can see that there is no periostome in here, there is no umbilicus here, there is no shape of the color, or the color is not has not been considered over here. It's all because that the shell, width, the aperture height, these are all quantitative characters which have been used over here. So we will be taking all those characters. We will select them, and then we will be doing our options. Options are fine. We are fine with this. And then we go for the OK. This is the result which we have obtained from our data, and this data clearly shows that we have these predictors. And summary of the classification: we have 139 total species of Macroclamis indica which have been identified, yet only 137 are correctly identified. Similarly, Xeropicta candaharica, 27 specimens have been were in total, 26 were correctly identified. In case of the Zooticus insularis, 15 were total and 15 were correctly identified. So it means that the summary of the classification shows that we are quite good in our identification. Similarly, 181 were the total number, and out of 181, 178 have been correctly identified. The proportion correct is 0.98, means 98%. We are fine with it. We are correct in our identification. Then we have, if you go for this particular thing, like we want to see which observation is should be reconsidered again, it means that there is 97, 118 and 141. 97 we have identified as Macroclamis indica, but it is the correct one should be the Xeropicta candaharica. Similarly, 118 has been identified as Macroclamis indica, but it should be identified as Xeropicta candaharica. However, 141 observation, that has been misidentified as Xeropicta candaharica, and it should be Macroclamis indica. So these three observations, through our results, we can again go back to those samples which we have put in our lab or somewhere like museum. We can go there and we can recheck them, and we can assess them again, so that we can confirm our identifications, whether these are hybrids or there are other cases, or these are some other things which may have happened, or rather it's truly the wrong identification. So we will have to check it again. So this is all about the use of discriminant analysis in the field of taxonomy. Thank you so much.